Introducing the xMAP SARS-CoV-2 multi-antigen IgG assay. The xMAP SARS-CoV-2 multi-antigen IgG assay uses multiplex microsphere-based technology designed to measure the presence or absence of SARS-CoV-2 IgG antibodies in human serum or plasma directed against the nucleocapsid protein, the receptor binding domain of the spike protein, and the S1 subunit of the spike protein. The assay can be run on all xMAP instruments, including the MagPix, Luminex 200, and FlexMap 3D systems. This instructional video will demonstrate the assay workflow and highlight the key steps to ensure a successful run. The xMAP SARS-CoV-2 multi-antigen IgG assay kit contains everything you need to set up the assay, including the microsphere mix, the detection reagent, wash buffer, 196 well plate, 2 plate seals, and the threshold card. You'll also need the separate USB, which contains the relevant product documentation and software analysis files. To begin the xMAP SARS-CoV-2 assay workflow, dilute each sample 1 to 400 by performing 2 stepwise 1 to 20 dilutions. First, pipette 190 microliters of wash buffer into the dilution container. Then add 10 microliters of non-diluted sample and mix thoroughly. For the second dilution, pipette 190 microliters of wash buffer into a separate dilution container and add 10 microliters of the sample from the first dilution container and mix thoroughly. If you are using the xMAP SARS-CoV-2 IgG control kit, dilute the controls 1 to 20 by adding 10 microliters of the control to 190 microliters of wash buffer and mix thoroughly. Once the samples and controls are diluted, add 50 microliters of the diluted sample or control to the provided 96 well plate. Then vortex the microsphere mix for 30 seconds. Add 50 microliters to each well and apply the provided plate seal. Next, incubate the plate for 60 minutes while covered at 25 degrees Celsius on a plate shaker set at 800 RPM. After incubation, transfer the plate to the magnetic separator for 2 minutes to allow the magnetic microspheres to separate. Then, aspirate the supernatant from the wells while the plate is still on the magnet. Be sure not to disturb the magnetic microspheres. Remove the plate from the magnet and add 150 microliters of wash buffer. Repeat the wash steps for a total of 2 washes, leaving the wells free of supernatant for the next step. Labs with higher testing volumes may consider using a plate washer for the washing steps. Please refer to the manufacturer's instructions for proper protocols. Once the second wash is completed, vortex the detection reagent for 30 seconds and add 50 microliters to each well. Seal the plate again with the second plate seal and incubate for 60 minutes while covered at 25 degrees Celsius on the plate shaker set at 800 RPM. After the second incubation, repeat the previous wash steps for a total of 2 washes, then resuspend the microspheres in 100 microliters of wash buffer. The samples are now ready to be analyzed on your Luminex system. To initiate the run, click on the Protocols tab at the top of the window, and then click Import. Be sure the associated exponent protocol is imported onto your PC. Then click OK. Next, select the Batches tab at the top of the window and create a new batch from an existing protocol, after which you will need to provide a name for the batch. Then select the protocol you wish to use and click Next. Once on the plate layout screen, click Eject. Place the plate on the carrier and add water to the off-plate reservoir, then click Retract. Select Run Batch to start the run, which will take between 20 and 60 minutes, depending on the number of samples you are processing and the xMAP system you are using. Once the run is complete, MagPix users will have to perform the MagPix serology post-clean routine. At the end of the run, you can analyze the data using the xMAP SARS-CoV-2 multi-antigen IgG assay software. To do this, open the software and log in. Navigate to the File menu and click Open. Choose the desired file, then click Open again. Enter the lot-specific values for the assay kit from the provided threshold card. Confirm the values are correct for analysis and click OK. Following analysis, the assay software presents a summary of the data, which can be printed or exported as a CSV or a PDF. For additional information about the xMAP SARS-CoV-2 multi-antigen IgG assay, please contact Luminex Customer Support or your local Luminex business manager.